So translation, the big picture of translation is that messenger RNA is translated into a chain of amino acids that could be called polypeptide. This polypeptide itself not always have a function, and when this polypeptide has a function, it's called a protein. In order to achieve a protein, we have to imagine like these amino acids are like Lego pieces. I think we've all played Lego or know what Lego is. So Lego pieces alone does nothing, but when you put some of them together in a coherent way, they could have a specific function. These functions, for example, the recognition of a site, of the recognition of a DNA portion, or we should imagine these proteins like having little arms and pieces or portions in this mass, portions where it could be like an arm to grab something and another arm to put them together, or we have to imagine like there are functional groups that could have a portion to interact with something else that could be another protein, or could be a DNA or RNA molecule, or could be almost anything inside a cell. This functional fraction of a protein is called a domain. So for example, we have in order to analyze proteins, they have decided to divide the structure into secondary, tertiary structure. We're not going to see, well, we're going to see this a bit further. So the main question here is how from a 4-letter language could be translated into a 20-letter language. How is that possible? So okay, we have these are our 20 different kinds of amino acids. As I told you before, these are like Lego pieces. We have the main structure of an amino acid, and in these pink boxes we have different structures that give rise to different features in them. We have different, for example, we have positively charged R groups and negatively charged R groups, which could give rise to this function, to these arms that I've been talking to you to. These Lego pieces that could perform something. We're not going too deep into amino acids. These are also the 20 amino acids. We have two kinds of abbreviations or symbols. One is a 3-letter abbreviation. For example, we have glycine here, the first amino acid. The abbreviation could be GLY, and we have its symbol that it's a single G. We have a lot of different properties here. For example, we have a hydropathy index, that is if it like the water, or if it doesn't. We have the currency in proteins that in a percentage. We have these values, pKa values, that is a value related to the strength of some bonds, some bonds that can be made in relationship to the pH, to the acidity of the medium. Well, I show you this just to know that these are different kind of pieces that have different properties and can achieve different goals. To answer the question on how does biology do, what does biology do, and how does it do it for translating from a 4-letter language to a 20-letter language, scientists have discovered something called the genetic code. In here we have two ways of showing this genetic code. For example, here we have the first letter, the second letter, and the third letter. Biology needs three different letters in order to can translate these combinations into at least a 20 letter code. So if we only had two nucleotides, we could have up to 16 different combinations, because it's been 4 squared, so it's 16. But if we had 3 letters in combination with 4 letters each position, we can have up to 64 different combinations. So 4 letter is pretty enough. I mean, 3 letters is enough to translate one message to a 20 letter language. So, for example, if the ribosome reads U, and U, I mean 3 times uracil, it will put a amino acid called phenylalanine. That stands for PHE, and so on. So we have in the left corner or side, we have the first letter. So when the first letter is an U, and the second letter is an U, and the third letter could be an U, a C, an A, or a G, we have this is just like the relationships from each combination of the 3 letters, which amino acid is it putting. So we have 64 combinations. Some amino acids have more than one, of course. For example, we have 4. These are called triplets or codons. So that's how we're going to call them from now on. For example, the UCU codon is translated into a SER that stands for serine. And for example, the GCA codon, what kind of amino acid does it put? I'll say it again, GCA. Can you find it? Okay, GCA is right here. So if we put a GCA, if in the ribosome the messenger RNA finds a GCA codon, it will put an alanine amino acid. That's what it is called. There are some important codons as the AUG. AUG is special because it is the initiation codon. This initiation codon puts an amino acid called methionine, and that's the point where the translation starts. Not all of the mRNA is translated. The ribosome starts reading the mRNA, and when it finds an AUG codon, it starts the translation always with a methionine that you can see here. And you have 2 kinds of stop codons, as you can see in here. AGA and AGG are marks for stop codon. So when the ribosome finds these kind of codons, it stops the translation. Here's another type of view of the genetic code, when we have the first letter at the inner part of the circle, the second letter at the outer or middle part of the circle, and the third letter in the outside. So we can find here, for example, the stop codon UAA. Yeah, so it's the same as we can see here. So that's the genetic code. And translation process, what happens or how does this process means? We have a 3-step process. First of all is the initiation, where all the parts are ensembling. The elongation, that is where the amino acid chain begins to grow. and the termination is obviously when it finishes the process, when it ends, when it finds a stop codon and terminates the translation process. We have many different kinds of molecules involved here. Obviously we have the messenger RNA with the initiation codon, because if we have RNAs without initiation codons, it won't start translating. We have of course the ribosome, that is a complex of a lot of RNAs and proteins and stuff. And we have the transfer RNAs, that is the one that does the translating process. It reads the codons and puts the amino acid where it corresponds. Also we have the initiation and termination factors, that are proteins that helps the ribosome to initiate and terminate the process. After we have a amino acid chain, the next process would be the folding and the post-translational processing. As I've told you before, the proteins can be changed, can be modified, can be altered or edited in a process called a post-translational processing. However, let's see a YouTube video. You know I love YouTube videos of translation. While I will be explaining it, I will explain it to you. So let's see the video. This is video made by a group called NDSU Virtual Cell, and it's about translation. So translation is the synthesis of a protein from an mRNA template. Yeah, that's what we're seeing here. So this process involves different kinds of molecules, including the messenger RNA that comes from a gene's transcription process, small and large subunits of the ribosome, and transfer RNAs and release factor. Here we're going to see three processes: initiation, elongation, and termination. As I said before, messenger RNA has a poly A tail at the 3' end, and it also have codons at the 5' cap. So we're seeing the poly A tail. These are the mRNA codons and the 5' cap. That is a methylated group. So as I said, the translation initiates when small subunit of the ribosome attaches to the initiation site. Afterwards, the transfer RNA or tRNA attaches to the AUG codon by recognizing this site named anticodon. This anticodon recognizes the site and pairs with it. At the opposite side of the tRNA, an amino acid is attached. In this case, it's a methionine, as I've told you before. That is always the first amino acid. Afterwards, the large subunit of the ribosome attaches, and the following transfer RNA recognizes, here comes the following RNA, it recognizes the codon and interacts with it, and the latter amino acid is transferred to the new one. This process is repeated several times, and this process is known as elongation. As elongation continues, more amino acids are attached to the string, and when a stop codon is found in the messenger RNA string, the release factor enhances the termination of the traduction. So we are still elongating the amino acid chain, and here comes the stop codon. The release factor attaches, and the amino acid is released. And well, that's it. We have our peptide or our protein, It depends if it has a function or not yet still.